I'm just got one. I don't have much to say, but Katie, when we were planning for this, wanted me to talk about kind of what's been funded. She wanted me to talk, but yeah, what's been funded in terms of therapeutic development, and also kind of what we have in the hopper, and where we are sort of negotiating here, probably for the next few years. Let me get this down. I all of a sudden don't know how to work the PowerPoint slides. Okay, so we're going to talk about a couple of different avenues. Some of the things you've actually already heard about today, and we're just kind of building and expanding on them. So just a quick diagram on drug repurposing for those of you in the room not familiar with it. Alicia already spoke to some of the therapeutics that she's using, but essentially taking available agents, either supplements or FDA-approved drugs, and testing them on disorders like MED13L. It's a cheaper way to go about it. It's already approved. We know safety, toxicity, efficacy, all of the things. And so it's really, it's kind of the faster way to do it. It is coming with a whole lot of other hurdles, but it is kind of what we have worked on, at least at the MED13L Foundation, pretty regularly, I would say, for the last, even before I started. And then just a quick slide on what those drug repurposing models are, and they're continuing to expand. And AI is certainly having a huge role to play in that because of the libraries that exist. And I'm going to talk a little bit about Unravel, which many of you in the room are participating with your children and their siblings, or you as a parent control as well. And that uses an AI-based model, but Alicia's obviously doing some drug repurposing. We've used University of Alabama, which mines the literature, and had a couple of hits come on there that actually has gone over to Alicia's lab to additionally test. Rarebase is on here as well. And I'm thinking of something else, but I can't actually remember. Actually, other people, other PAGs. So these are the therapeutics that we've really talked about in the last year, and I just wanted to give a quick update on where they are. Naproxen, which is what came out of Rarebase. This is actually was shown to upregulate MED13L mRNA. So again, we're speaking to haploinsufficiency. So one good working gene, and it's a loss of function. Working with Jen right here, down here. Nope. Who tries to ignore me, but she can't hide, on sort of next steps on that. Naproxen is obviously diffusely available. It does come with, risks in terms of side effects, especially the GI irritation, stomach irritation. It has been studied in kids, but we're really looking at, there probably is a very strong placebo effect, because in and of itself is an anti-inflammatory, right? It's meant to make you feel better, regardless of if you have MED13L or don't. And so those are the things that Jen and I have started having conversations on, and that you've heard a lot about today, which is around outcomes, outcome measures, and how do we measure that and differentiate it from, something you think is happening versus something that is actually happening, along with monitoring the side effects and the risks that we don't know that would come with long-term use of a therapeutic like that. Phenylbutyrate is something that we funded with the Caddy Kang lab, and we're looking to further pursue that after we have a bunch of neurons growing right now, which is very exciting, and it was a huge gap. And so once those neurons are finished growing and being characterized, this is something we'll go back to Caddy with. Caddy's done this in SLC6A1 Connect. She's done it with a bunch of GABA variants, as well as SYNGAP1. So we think there's probably something there that comes across. She has a very early signal of phenylbutyrate working in some fibroblast cells. And so we really want to make sure that we're validating that in MED13L neurons. One of the issues with phenylbutyrate is that it's extremely expensive. It is available on the market. It's just under $1 million a year. So getting that through a payer system, yes, that's right. Small potatoes. Getting that approved, through insurance is obviously a huge challenge. And so I've been working really closely with Amber Freed and SLC6A1 Connect on thinking about a therapeutic alternative or an accessible product. N-acetyl-DL-leucine or N-acetyl-L-leucine or NOL or Tanganil, all of the above. Lots of you in this room are probably on that Facebook group. This was something actually discovered by SYNGAP1, has a really sort of broad mechanism, and we do have a lot of parents whose children have tried it with some, and MED13L data that I have seen from what you all have reported looks to be pretty helpful and very well tolerated. It is was only getting it through importing it from France, and we were no longer able to do that. So access has become an issue. So still working on that. So this is what I mean by the barriers that come with like discovering drugs that are already available and like being able to implement it in practice. So you get a positive result, and then you've got 14 more steps to move forward and 5 to come back and 14 to move forward. So, and then Alicia's name has already been mentioned like 100 times today. So we've already funded for Dr. Randy Strick's lab and now Dr. Alicia Campbell over $120,000 in grants for mitochondrial drug repurposing for the lab tech that she mentioned today, as well as herself, to continue testing that. That funding goes through the middle of next year. So we still have a lot more things to learn and find out about the work that they're doing. Just throwing Unravel up here real quick. So those nasal swabs, if you're participating in across the hall between your child and either a sibling or your child and a sex-matched parent. So it's an interesting model. This is Richard and Frederick were here, and I think Fred's coming tonight to City Point. Oh yeah, he's standing up in the back. From Unravel, they're here local to the Boston area. And this is just an interesting and unique approach to now figuring out, like, how do we multiply this across a patient population? And so they're using nasal swabs to map the transcriptome and then putting that up against the LINCS library, which is a 40,000 drug library, and using some of that approach to get some drug hits. I've seen these hit lists. They are massive and have some really interesting data and information. We are also have been able to negotiate this with Combined Brain. So the goal is actually not just to look at MED13L, but similar to what you heard Anna Fauser talk about this morning with proteomics projects, to actually look at MED13L across the spectrum of the neurodevelopmental disorders that are participating in this project throughout the year. So this will take some time to get out because the data set's going to be so big, but it also has the opportunity not just to think about agents that might work for MED13L, but agents that might work for MED13L or agents that work for something and MED13L. So really to broaden what our population looks like and gives us a broader toolkit and opportunities to combine other drugs to make some improvements in everyday life. And then we just heard Ryan talk about some gene-based therapeutics and the upstream open reading frame. We actually did, in the same time that we were funding Ryan, we did a quick hit with using some microRNA blocking technology for ASOs with RNA Spire, which they also use IDT to get their ASOs. And Mozzie Therapeutics. And we actually, so this is a shot on goal. And sometimes in science, your shots on goal don't pan out, you miss wide. And so this is a case of where we actually did look at this. It wasn't that expensive. And so from a board decision, they made the idea to go forward with this. It's about $35,000. They were able to get about 10 micro RNA ASOs, test it, in generic neuron models and found no effect, found no change. And so they repeated that not only in these SHSY5Y models, but also did it again in another generic neuron model and found the same thing. So this is something, right? We threw a ball at the target and it just missed. And so that's, when we're doing drug development, we're going to miss. We're all going to miss. But I think it was Wayne Gretzky who said you miss 100% of shots that you don't take. And so this is one, and that's okay. But it also tells us what doesn't work. And so that can be just as valuable as learning what does work. And then these are the last two things that we're looking at right now and negotiating with, which is in the Nadav Atuv lab. This is another gene-based therapeutic, CRISPR-A. So not kind of what you've heard today in terms of like prime or base editing. This is an upregulation. It has the potential to work across the entire haploinsufficient MED13L population. And then the other thing is the Jeff Collier lab, who's at Johns Hopkins, and that's using this poly-A tail mimetic, which again just elongates the tail, so it allows it to keep coding. And so those are two conversations that we're having right now and negotiating with in terms of funding to continue to move forward in our therapeutic drug development. So that's my quick update. I have to throw out the priority survey because this is what matters and we're not going to get anywhere. And I can't have a conversation with Jen about how we're going to do outcome measures if I don't know. Yeah, you got to tell us. And so please fill out the QR code, stop by the registration desk. It'll be there as well. And then I'm just going to let Katie close us out because we're running behind. Yeah, all right.
While she's pulling up the slides, totally forgot to go over this morning. On the back is your QR code, one to make donations to keep all this amazing work that you heard about keep going, and another one is a lot cheaper. It's your photos. So scan this, and I'd love to just upload everyone's photos. And I think you guys are probably sick of seeing like the same 10 photos we have from the 2022 conference. So give us more material to work with, and let's take some really fun photos tonight at our party because you guys seem fun. Hopefully.
Can you keep on talking?
Yeah, I'm going to keep talking. All right. So I just finished this about 10 minutes ago, so I don't have it memorized. But I'm going to start by treating this like my Oscar-winning moment, and I'd like to thank everybody who made this happen.
The music's already playing.
Is it the wrap-up music?
Yeah.
You guys are all probably feeling that. But
where's the cowbell?
Our volunteers, our sponsors, our researchers, especially some of the researchers that haven't received a single dollar from us, you can scan the researcher bios and kind of figure out who that is. And buy all the researchers a drink tonight, but especially the 20-year-olds who are doing all this for free. So
no, don't buy 20-year-olds drinks.
21. It was a test. You passed.
Like, give them a side sip, but like we're gonna like keep it legal.
Every party has a pooper. So this is, a picture of some of the people that got us here. The middle picture is from our West Coast meetup. I didn't have time to put together some of the other meetups that you guys threw. That was so awesome. I'd love to just keep that going too. That's Vanessa and I at Global Genes, just trying to learn as much as we can. This is Rachel and I with the RareX team. But most of all, I want to thank you guys. Like, showing up, I actually can say I know what a sacrifice it was to get you guys here. You're financially, logistically, you chose to spend your vacation with us, and it means the world. And that's how I know we're gonna do stuff. We're gonna make this happen. Your presence is powerful. Like Rachel said earlier today, as parents, we all remember the day when our lives changed. And we realized our parenting journey was gonna be different. There's a poem that actually Michelle shared when I first joined and I signed into a Zoom, and it's about, arriving in Holland. Other people I've since learned call it Beirut, and I think that also kind of resonates a little bit. But we also remember the day we made the decision that this isn't enough, not for our kids, and we decided to make the change. And this foundation was born from that drive. And I just want to share a quick story of how I got here. April 18th, 2022 is when my world shifted. A doctor sat across from me and told me my son had a disease so rare, never heard about it before, didn't know much about it. He told me letters, numbers, that was it. Okay, what are the treatments? What are the therapies? I don't know. But he said, there's a Facebook group. I'm like, woo! I gotta get back on Facebook. I just got off Facebook to protect my peace. And if there's any genetic counselors here, stop. Oh, he was like, and you're gonna come back and you're gonna teach me. I'm like, well, you can give me my copay money back. Stop. Tell your Tell your geneticist. Stop saying that to us. We don't want to be the teachers. Like, I don't want that. You're the doctor. So, I kind of sat with this, and I ran to the internet and I saw, developmental delays, absent delayed speech. And I looked at my 16-month-old and, my dreams of, Little League, going to college, first dance at his wedding, were kind of slowly transitioned into like daily therapies and being a lifelong caregiver. And in that moment, I felt as a mom, like my power slipped away. So I joined that Facebook group. And I saw all of you, and I saw your stories, and I saw your conversations, and it gave me hope. But I also saw your questions. And, I looked at Colin, and I was like, let's get to work. So I joined the foundation. So actually, I had a really good cry, like a really good one, whew, that was great. And then, I kind of metaphorically put it in my handkerchief, put it in my pocket, and I let that drive me every day. Sometimes you gotta take it out again. You gotta allow yourself that good cry. But I joined the foundation. The foundation joined Combined Brain. We tricked Rachel into joining us. Rachel, did a strategic plan, got it published. We raised money, and we funded our first studies that you guys heard about. And I know someone asked earlier, kind of what is that price tag? And Rachel and I were just kind of doing back of the numbers, crunching really fast. And to date, we've spent around $435,000 on the research you guys heard about today. And to kind of keep it moving and to fund some of those options you saw, we need to turn that number to a little over $600,000. So we launched RareX. We got our gene review published by Dr. Alicia. We completed our disease concept model that you heard about that's about to be published. We discovered many of our kids have apraxia and dysarthria, and we published that too. And now we've identified drugs that upregulate MED13L. And so now we're preparing for what many of us have really hoped for, a clinical trial. But we can't do trials without data. And so that is why I have been bombarding you with RareX, and then I made Inan bombard you with RareX, and then I made Michelle reach out to you and some of the other regional ambassadors, and this is why. We asked, and over 100 of you guys showed up and completed those brutal surveys, especially the Vineland. But because of it, we are going to have the most comprehensive study on MED13L ever published. And that is going to directly change the care our children receive. You did that with your time, with your honesty, and with your story. You are the engine of this mission. Fast forward to today. And yesterday, we started the EEG study, and nearly 30 kids, hopefully, there's still a few extra spots in case anybody wants to sign up, go up there, with research-grade EEGs and a world-class scientific team searching for a biomarker that we can show when a drug is working. That costs around $40,000. Because there are drugs that can help our kids live their best life, But as we heard, the FDA needs us to prove how. So today we're launching the MIND study, a natural history study with Boston Children's. It's $250,000. This is our biggest investment to date. And we've just launched Citizen, our medical data platform, because we need both pieces of the puzzle. We need caregiver-reported surveys, which is RareX and Simons. Thank you guys, because I know it's brutal. And Citizen, so much easier. You guys just do it, you can upload it. So if you can do only one thing from this weekend, I would say scan Citizen. It's actually, you need to show me proof to get into the party tonight that your sign up thing. So I will be checking, won't be checking IDs, will be checking this. So please scan this. And you also heard we're launching a community survey because everyone in this room is different. We come from different backgrounds, values, and we all have different goals for our children. Some are focused on therapeutics, and some are on acceptance, inclusion, and quality of life. And all of it matters. All of it is valid. No matter your goals, cure, care, quality of life, the foundation exists to support all of them and the community we're building together. I've read your posts, your emails, your texts. I've cried with you, and I've celebrated your inchstones with you. And I felt the weight of the unknown alongside of you. This is why this survey matters. So scan this one, guys. What do you want us to go after next? What would make the biggest difference in your family's life? This community belongs to all of us. Your voice shapes the path forward. I've heard a lot of feedback more than once. I personally am confusing. I understand that. ChatGPT is helping me. And all of it, it's a lot. Katie, it's a lot. Stop. And I hear that. I receive that. And I am here for you. I'm here to answer your questions. But I also need you. I need each and every one of you. You don't need to be a geneticist. You don't need to run a nonprofit to make a difference. My last biology class was in 2002, and I got a C plus, but still. And now I'm just a lawyer who reads small print for a living. I didn't totally understand everything going on. Rachel does, so. But, we have Vanessa, and not everyone can be a Vanessa, who's an amazing nurse and insanely talented with her graphic design. And Rachel, who I don't even think there's words to describe Rachel. And I think we all really saw that with the materials that were put together today. But you don't need to know how to explain the research we're funding. Ask a local restaurant to donate 20%. Invite your friends and family. Start a Facebook fundraiser. Share our website. There's no Hallmark card for what we're going through, but your people want to show up for you. The small act gives them a way to help, and it helps us grow, and it helps us bring in the resources we need. And I know how hard it is to fill out all of these surveys and have to answer no, no, no. And that's why I use a mantra I hold close. Not yet. And it's the power of yet. And that's why you'll see it on some of the bracelets out there. My Colin cannot speak yet. He cannot run or jump yet. There is no treatment yet. This is how we take back our power, by making every story count. I have so many hopes for this weekend, but my biggest one is to build community. We need more parents helping shape our goals, research, education, funding, outreach, and more. Everyone wants something different, and we need to make it all happen, but we need you. Sometimes making the world better doesn't mean reinventing it. Sometimes it just means creating space for the people already in it. Let's imagine a world worthy of our children, of their love, kindness, and joy. And let's make it happen. Let's lead with love. Let's keep learning, even when Google tells us we're wrong or no one knows the answer yet. Let's not be afraid to make mistakes and not be afraid of missing the lessons they offer. We're not the future. We can't wait for it. We are the moment. And we must use our voices for our children who don't yet have one. We've heard no or I don't know so many times. It's become the soundtrack to our yes. We're not waiting for a turn. We're stepping up and saying, it's our turn now. Thank you for being here. Thank you for trusting me. And thank you for making every story count. Let's get to work, guys.